This is the first video in the series of gene sequencing, and today I'll be explaining the protocol for the very first step, which is DNA extraction. Before we can run any PCR, gel electrophoresis, or sequence the DNA after, we require to extract that DNA from the amniotic fluid or the blood of a patient. Okay. And this step begins once we acquire blood. So this EDTA blood is this vial, generally has a purple cap, and the blood has been extracted and is kept in EDTA, which we know is a buffer. This provides isotonic conditions, right? And we'll set this in a tray. Okay, once you acquire it, you label it, because it's very essential. Any sort of mislabeling, losing track of it will lead to misdiagnosis. So we note it down in the registry, and we write it over here. We label it with a permanent marker, and we allow this to sediment. Sedimentation here is essential so that there will be a gradient of the blood here. And why this is essential is because when we extract this DNA, we pipette out DNA, and we take this in our labeled vial. Okay, the same label here, we'll have here a numerical one, and to extract the DNA we require a certain group of cells. So general knowledge, what is the blood made up of? Okay, we know there's plasma, there's WBCs, there are platelets, there are RBCs. Okay, major components. What we are targeting are the WBCs. Okay, these leukocytes, because RBCs do not have a nucleus. A lack of nucleus means lack of genomic DNA, and the genes we are targeting will be there in the DNA, right? So this DNA extraction we're doing, we're trying to get the WBCs, okay, and we want a good amount, okay, because the more DNA you get, you can run various processes. One DNA extraction can be used several times. Sometimes you don't get a definite result and you again need it, and you wouldn't have to go through this entire process if you get a good amount of DNA. So when sedimentation happens, you see three layers. Okay, there's this darker one, by density it has sedimented, which is RBCs. There'll be a buffery kind of layer, and the top layer which is more liquidy. Okay, and the one we want is this buffery layer, okay, in the middle, the junction here. So we pipette it out at the bottom of this junction. Okay, so we'll target that buffery layer, and we'll take 200 microliters of EDTA blood. Okay, always use separate tips. Do not mix, or there will be cross contamination of blood samples. Okay, and for disposing we use sort of a Ziploc bag, right? Because we have to be careful and wear gloves. Anytime you see a droplet, you switch gloves. Now we'll add proteinase K. Okay, the K here stands for keratin, which we know is an essential amino acid. Proteinase, like DNAs, RNAs respectively, what do? They digest proteins. RNAs digest RNA. So This is a protease and it digests most proteins and even tough ones such as keratin. So we are adding this here and we touch mix the solution. Touch mixing is vortexing. It is done. There's this device where you touch mixes just for about 30 seconds. We proceed by adding 100 microliters of lysis buffer and we touch mix this more thoroughly. You'll see that this starts to appear blackish. Okay. Initially when you touch mix it gets a little brownish, it gets darker, but when you touch mix here again for about 2-3 minutes it turns blackish. The reason being, now when we took the sample there was a mix of mostly more of WBCs but also RBCs. Of course there's no complete separation, so the lysis buffer lyses the cells, right, and that leads to release of hemoglobin, and this hemoglobin gives this blackish appearance to our suspension. Next step, we'll add 200 microliters of ethanol. Okay. So this is now done using a column. You get these columns. Okay. This has pores, right? Small pores. And this column is white. The one used in gel electrophoresis is yellow. So we add ethanol and then we add our DNA, which was in this vial, right? Okay. This had been kept in a water bath. Okay, there's a dry bath at 56 degrees Celsius set for 10 minutes. We incubated that. Okay. That gets the lysis buffer working. So the lysis is happening and complete lysis is happening, and now from this we pipette out all of the DNA, okay, all of this blood sample, and we add it here. After that we're going to run the centrifuge. Okay. The centrifuge will be run at 12,000 RPM for 2 minutes. Okay, what is a centrifuge? This machine with the rotors, we use centrifugal force to create a density gradient and separate our materials. So here the ethanol and the lysis buffer, all of this is gonna get separated out and we will be left with DNA along with still some of this reagents we use. The DNA is yet to be purified, and that's what we are going to do in the next steps. Okay, running the centrifuge also you have to keep in mind that the sets of tubes you keep are equidistant. Okay, so let's say you keep 4 here, you ensure the distance between these two sets is equal for the proper working of the centrifugation. And let's say you have an odd number, right, 7, so you take one blank, just pick up a tube and keep it there so that there's a balance between the two sides of this rotor. Okay, Anyway, moving on to the next step. After you've run the centrifuge, what you do is discard the supernatant. Meaning this column here, you throw out this tube. Okay, this is separated. There's this, and this has been, put inside this column. We throw this out, and we keep a new tube here. Okay, so new tube is kept here, and now we add our wash buffers. Okay, we're adding 500 microliter of wash buffer. This wash buffer 1, we run it again, we centrifuge it, same RPM for 2 minutes, then we discard the supernatant. We have to now again add another 500 microliters of wash buffer 2, right? So no need to throw away the tube, simply we take this tube, right, hold this in your one hand, hold this in your other, just pour out all of the supernatant into your waste basket and just put it on top again, and then add your second wash buffer, centrifuge it this time for 5 minutes, then we give it a dry spin. A dry spin means you're going to simply throw away your liquid here and just run this dry. Okay, let's be sure to know what each reagent is doing. Ethanol here helps in precipitation. It neutralizes the charges and it makes the DNA insoluble so that it will precipitate. Now after the dry spin, we keep it on a 71 degrees Celsius dry bath. Okay, and this is to ensure that all of the alcohol completely evaporates. Okay. So we have, after this step, we have thrown out the tube, right? And this is in this new tube here, and from this column all of the alcohol is evaporating. You're not closing the cap, of course, and this will take about 15-20 minutes. After that, you add elution buffer. Elution buffer, now your DNA is in this part of your column. We want it here, right? So separated here, you can't see it, of course it's too tiny, but we add this elution buffer about 50 microliters, and then we incubate it at a room temp, right, for about half an hour, so that it just soaks up there. And the working of elution buffer is at room temperature, and then we centrifuge it for 2 minutes again, and there you'll have it. You'll notice we're going to throw this out, and you'll see here will be a nice amount of liquid. The more there, the better. And now you can store it. If you have to use it, you use it, or you store it in minus degrees. Keep it there. If you keep it outside, there's no damage. Ensure that the label didn't get wiped off, rubbed off in any of the process. Each step keep checking, because you can't mess that up. And well, anyway, you can keep it at room temp because DNA is stable, unlike protein and RNA. This was the first step. In the next video, we'll discuss normal PCR steps and sequence PCR. and the purification after.